For subject botany, SEM, SEM 6th topic is non-cyclic photophosphorylation. Non-cyclic photophosphorylation. So friends, good morning. Today I am going to explain about the non-cyclic photophosphorylation. Who expressed this non-cyclic photophosphorylation is expressed by Armon. So in the presence of the light, in the presence of the light, the electrons of PS1 and PS2 in the chloroplast, they transverse through various electron carriers which are present on the thylakoid membrane. So these carriers does not reach the pigment system. So the electrons does not reach the pigment system. So this is known as a non-cyclic photophosphorylation. So this non-cyclic photophosphorylation it is discovered by Armon. This is discovered by Armon. So non-cyclic photophosphorylation is discovered by Armon. So here in this non-cyclic photophosphorylation, the electrons they can reach to the electron receiver NAD and FAD. NAD. So nicotine amide dinucleotide. So this is the electron receivers. So the two electrons plus two protons in the presence of NADH dehydrogenase, NADH dereductase, then immediately it can reduced as a NADH plus H plus. So this is the reduced NADH dehydrogenase. So these forms are formed in this way. So the electrons which is excited, they are released from the exciting stage of the pigment system 1, and the pigment system 1, the deficiency of the electron at the pigment system 1 is fulfilled with the electrons which was released from the pigment system 2, and the pigment system 2 electron deficiency is fulfilled with the from the electron which is released from the magnesium and chlorine ions. They can transfer the electron from which are released from the photolysis of the water. So this is the non-cyclic photophosphorylation theory. So if you can see the non-cyclic photophosphorylations diagram. So actually here what is that? So pigment system 1. So pigment or PS1. So in this PS1, what is the reaction center? P700. So P700 is the reaction center. So it is nothing but a chlorophyll A pigment. P700 is chlorophyll A pigment. Why we are point out the P700 as, because the P700 is the 700 is the photo 700 nanometer wavelength of the spectrum. So this chlorophyll pigment, it can able to receive this much wavelength of the spectrum. Hence it is named as a P700. But it is a chlorophyll A pigment. And it receives Okay, it receives the photons. Okay, these are the photons. So the wavelength they reach the photosystem 1. So among from this photosystem 1, one chlorophyll pigment, any one chlorophyll pigments, they can receive the photons of the radiation of the wavelength. Then they will stimulate and they are excited. They are excited. These chlorophyll A pigments, they are excited and they release the electron. They release the electron, and this electron is received by FES protein. Fe ferrous sulfur protein. So the primary electron receiver in PS1 is FES protein. So this FES protein again it transfer the electron to ferridoxine. So these electrons, they are transferred to the ferridoxine. So this ferridoxine is transferred to the electron to NAD minus plus 2 electrons plus 2 protons. So gives rise what will form it? So NADH plus H plus are forming. So this is nothing but a PS1 for pigment system 1. So here, so photon is came and that photon is received by pigment system 1. Then pigment system, in this pigment system 1, the chlorophyll pigment A, which is called as a reaction center, so that is P700, is excited and is releasing electron. So that released electron is received by the FES protein. So the FES protein that the electrons receives, and that electron again transferred to the oxidized ferridoxine. So that ferridoxine in the presence of the ferridoxine NAD ferridoxine reductase, in the presence of that enzyme ferridoxine NAD reductase enzyme, in the presence of that enzyme, that ferridoxine that transfers the electron to the oxidized NAD. That oxidized NAD that receive 2 electrons from the ferridoxine and 2 protons from the stroma or the lumen, stroma of the thylakoid or chloroplast. So 2 protons received from the stroma of the chloroplast, and then it is reduced as a NADH plus H plus. So this is something about the what is the PS1 system. So here electron are released and electron deficiency is occurred here. So how the electrons are transferred to the P700? So here next one is PS1, this is the PS1, then second one is PS2. So PS2, and in this PS2 the electron, the reaction center is P6CIT. So P6CIT is the reaction center. So it is also chlorophyll A pigment. P6CIT is the chlorophyll A pigment. So it would like to receive the P6CIT nanometer wavelengths of the spectrum. Hence it is called as a P6CIT. So it can act as a reaction center in pigment system 2. So in PS1 chlorophyll A is very very more in this pigment system, but in PS2 chlorophyll B is more than chlorophyll A. So here again the wavelengths are reaching to the PS2. So photons, so these are the photons. So photons, the internal energy of the photon is called as a quantum. So with that quantum energy is transferred to the P6EIT. So then immediately the P6EIT reaction center, it is excited and it releases one electron. Then in this one, the electron receive the PO fighting. The electron receiver is PO fighting. Okay, the electron receiver in PS2. Electron receiver. So the electron receiver in PS2 is pheophytin. So pheophytin is also chlorophyll A pigment. So it is a colorless pigment. Pheophytin is also chlorophyll A, but it is a colorless pigment. So it would like to receive the electron from the P6EIT. So this P6EIT again it transfers the electron. It transfers the electron to the quinone. So this quinone electron it transfers to the quinone. That quinone transfers to the cytochrome F. Then cytochrome F to the plastocyanin. The plastocyanin it can transfer the electron to the PS1. So if you can see the plastoquinone, in the past class I told plastoquinone it has a cyclic nature. So the plastoquinone, when it receives the electron, it can modify it as a semiquinone. It modified as a plastosemiquinone. So plastoquinone, it receives the electron. So it receives the electron and it can form as a semiquinone. Okay, the semiquinone again it receive two protons from the stroma of the chloroplast. So here two electrons. These two electrons are transferred to the quinone. Okay. So two electrons, they received the two electrons from the pheophytin. So this pheophytin transferred two electrons to the quinone. Then it immediately this quinone received the two electrons and it will be changed as a semiquinone. So this semiquinone again it receives it takes two protons from the stroma of the chloroplast. Then it is formed as a plastoquinol. PQH2 plastoquinol. So it is plastoquinol. Then in this plastoquinol, then it is again oxidized into plastoquinone. So in this plastoquinol, it releases two electrons and two protons. So these two protons they transfer into the lumen thylakoid lumen. So they transfer to the thylakoid lumen, but these two electrons they transfer to the where cytochrome F. Cytochrome F. Okay, cytochrome F. This cytochrome F they receive the electron from plastoquinol, and this electron again that transferred to the plastocyanin, PC, plastocyanin. So this plastocyanin, this receive the electron, and that plastocyanin it directly transfer the electron to the P6EIT, which is present in the PS1. So this is something about the something about the non-cyclic photophosphorylation. Okay, here the deficiency of the electron at P700 is fulfilled from the P6EIT. So next, here also electron deficiency is occurring. So the electron deficiency which is occurring at the PS2, it is fulfilled from the thylakoid lumen. So this is the thylakoid lumen. So in this thylakoid lumen, the thylakoid lumen is completely fulfilled with the water and ions, water and minerals. So near to this PS2, magnesium chlorines ions are arranged. They are situated near to the PS2. So in this lumen, so for example it is a lumen, so in this lumen water is there. In the presence of the water in a photo, in the light, okay, during the daytime, in the presence of the light, that water molecules are photooxidized and they release 2 electrons and 2 protons and of oxygen in the light only. So this is called as a photolysis of the water. So it was explained by the Robertson. So Robertson, it was explained by Robertson. So it is Robert Hill, not Robertson. So it is explained by Robert Hill. Hence it is called as a Hill reaction. It is also called as a Hill reaction. So here, for example, this is a lumen, thylakoid's lumen. In thylakoid lumen, this is one H2O. So one H2O molecule, it is oxidized. So it is photo-oxidized into 2. So so here 2 electrons, okay, 2 protons and plus 2 electrons plus of O2. So like this one, if we can 2 molecules of the water, it can releases 4 molecules of the proton, 4 molecules of the proton plus 4 electrons plus 1 molecule of oxygen. So here what is happening? Here oxygen is evaluating by the photolysis of the water. So the oxygen which we are taking inside, so that oxygen is releasing through this photolysis of the water. And these electrons, so these electrons with the help of the magnesium and chlorine ions, these electrons which are released by the photolysis of the water, so that electrons are they transferred to the P6EIT of pigment system 2 with the help of the magnesium chlorine ions. So this is the deficiency of the electron fulfilled by this procedure. So this is called as a non-cyclic photophosphorylation. So here what is happening? So here one thing, so one ATP is also formed here. One ATP where during the quinone valium, in this period, what is that? So during the electrons when transferred to the cytochrome F, at that time one ADP plus inorganic phosphate then it is forming as a ATP. So here one molecule of ATP is synthesizing in this non-cyclic photophosphorylation, and here 2NADH is forming here in this non-cyclic photophosphorylation. And also in this non-cyclic photophosphorylation, so the non-cyclic photophosphorylation actually the non-cyclic photophosphorylation, it is discovered by Amon, and it is discovered by Amon in higher planes, and the Hill and Benda they proposed Z scheme involving the photophosphorylation. The Z scheme of the photophosphorylation is proposed by Hill and Benda. Next, in non-cyclic photophosphorylation, the PS1 and PS2 pigment systems are involved here. Here, the photolysis of water takes place. Photolysis of the water takes place, and oxygen gas is released in this one. Photolysis of the water is taking place, and also oxygen is releasing in this way. So the present the process of beginning of the absorption of light, it is by the PS1. So the reaction center P700 is excited, and it releases 2 electrons. These 2 electrons reach the FES protein, and then they reaches to the oxidized ferridoxine. So the ferridoxine gets the reduced, and NADPH is formed in the above reaction. As the electrons from P700 reaches the NADPH, the deficiency of the electron occurs at P700 reaction center. Then this deficiency of the electrons at P700 is obtained from the P680 of PS2. So here the PS2 absorbs the lower wavelength. The PS2 is absorbed the lower wavelength, and the reaction center P680 is oxidized, and 2 electrons. Not oxidized. So the PS2 is excited. P680 is excited and releases 2 electrons. These 2 electrons are released from the P680, and these electrons first reaches to the pheophytin, and a deficiency of electron from photooxidation of water, and electrons are removed from the water by the magnesium and chlorine ions in the presence of the hydrogenesis enzymes, hydrogenase enzymes. So the protons, 2 protons and 2 electrons, 2 protons, 2 electrons, and of oxygen are released. The 2 electrons they transfer to the PS2, and the electrons from pheophytin reach to quinone. Then the quinone gets the reduced and reach plastoquinone, and the plastoquinone receive the plastoquinone receive the electron, and non-cyclic electron transporter. It contributes 6 protons to the lumen of the thylakoid. So here the 4 protons from the quinone cycle, 2 protons from the oxidation of the water. So the electrons are transferred to the plastocyanin. How here one NADPH is formed. One NADPH is formed. So the NADPH is from the plastoquinone to cytochrome F, 1 ATP plus the plastoquinone to cytochrome F, 1 ATP is synthesized. So here one ATP is formed, and also two NADPH are formed. So in photophosphorylation, 3 ATPs and 2 NADPH are formed. So the energy formed in high plants, are is utilized in dark phases. What are the dark phases? So with the help of this NADPH and ATPs, the dark phases are utilize this reduced energy, and they synthesize and they convert the carbon dioxide into the carbohydrate. So this is something about the photophosphorylation. So for one carbon dioxide molecule, from one glucose molecule C6H12O6, 3 ATPs and 2 NADPHs are required. So this is something about the non-cyclic photophosphorylation. Thank you very much. Then we will meet in next class. Thank you one and all. Thank you.